Hello, and welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the final episode of Plantopia for 2024, and today I'm delighted to welcome Dr. Lisa Castlebury, research mycologist and research leader of the Mycology and Nematology Genetic Diversity and Biology Laboratory with the U.S.D.A. ARS Beltsville Agricultural Research Center in Beltsville, Maryland. Dr. Castlebury completed both a BS and MS in biological sciences at Western Illinois University and a PhD in plant pathology at the University of Illinois, Urbana-Champaign. Dr. Castlebury completed her postdoctoral work with the FDA at the National Center for Toxicological Research in Jefferson, Arkansas. In 1997, she began her long career with the USDA as a research associate in the Systematic Botany and Mycology Lab. She then served as a plant pathologist in the USDA APHIS in the National Germplasm Quarantine Center. In 1999, she accepted a position as a research botanist mycologist and would work her way up the ranks over the next 2.5 decades. In 2014, Dr. Castlebury was appointed acting research leader of the Systematic Mycology and Microbiology Lab and director of the U.S. National Fungus Collections. In 2018, she was appointed acting research leader for Mycology and Nematology Genetic Diversity and Biology Lab. Her recent work has focused on digital imaging of specimen information for the U.S. National Fungus Collections, identification of fungal intercepts at points of entry, DNA barcoding, and developing diagnostic reference tools for plant pathogenic fungi. Dr. Castlebury is author to almost 200 research publications, including 24 first reports of disease. In the late 1990s and early 2000s, she was among an important cohort of mycologists doing some of the first DNA-based work on the Diaporthales, Stachybotrys, and rust and smut fungi. Dr. Castlebury has mentored a dozen postdoctoral associates and co-advised numerous grad students. She is an active member of MSA, APS, and AAAS. It's my pleasure to welcome her here today. Dr. Castlebury, thanks for being here.
Yeah, thanks for the invitation.
For sure. So you are a botanist and a research leader of Mycology and Nematology Lab.
My official title is botanist. However, I'm a little hard-pressed to identify a lot of plants. There is no such thing as a mycologist title in the USDA, and botanist was the closest they would get at the time.
That's likely due to the fact that a lot of mycology programs were in botany departments for a long time, correct?
I think that's true, and for a long time, it was thought that fungi and plants were very closely related. It turns out fungi and animals are a little more closely related, so.
Absolutely, and that makes sense. And when I saw that title of botanist on there, I thought, I wonder if that's a hat that the USDA put on you. And really, by all accounts, you are a mycologist and a plant pathologist. And one just needs to quickly look over your research contributions over the last 30 years and realize how much mycology you've done in fundamental fungal biology and fungal ecology. One of the things that I was excited to talk to you about today is that the timing of your career really lines up perfectly with the coming of age for DNA-based studies of fungi. You finished your PhD at the University of Illinois in 1994, and shortly thereafter, you went to work for the USDA ARS.
Right. I had a short postdoc, a couple of years with the Food and Drug Administration lab in Arkansas. And then, as funding, of course, is never completely stable for postdocs, I went to the USDA in Beltsville, Maryland.
And what did you work on when you were a postdoc then?
I was recruited specifically to work on carnal bunt of wheat, Taletia indica. At the time, it was thought that it was found in Arizona. And this is a big quarantine problem because most of the countries in the world were quarantining against carnal bunt. My job was to find out what the host range was from morphological examinations.
And I assume that proved to be a little bit challenging based on the fact that a lot of rusts and smuts, they tend to have very similar looking resting spores.
They do. It is a little challenging to distinguish them morphologically. It turns out that in Talicia, a lot of the species are relatively host specific, let's say, and the spores that were causing the most trouble had been found associated with ryegrass in pasture mixes. And it turns out that no, they weren't Talicia indica or carnal bunt.
Okay. And before going forward to talk about your long career with the USDA, I thought we'd actually go backwards in time a little bit. I had mentioned that you went to the University of Illinois at Urbana-Champaign. I think you were at Western Illinois before that.
I was. Yes, I was.
Was that for your undergrad or was that for a different, your master's degree?
Both. It's the Western Illinois University is very close to the farm where I grew up. And they offered me scholarships and things. Seemed like a good place to go. So I did a bachelor's degree in biological sciences and then a master's degree in biological sciences. But my focus was microbiology, really, in both cases.
And I think I saw you worked on the Pleosporellian marine fungi for your master's. Is that correct?
It was a marine fungus. I was looking at Dendrophiella was the name at the time. I think since then it's had at least one name change, if not more. My master's degree advisor had done his postdoc on marine fungi. So he had a large stock of these in his refrigerator, and I was looking at levels of protein and DNA and RNA being produced in different types of culture.
Hmm. And you said you grew up on a farm not far from where you went to, as an undergrad. Was this a working farm?
Yes, it was and still is.
What kind of farm was it?
At the time, and I guess still, it was corn, soybeans, forage crops for the cattle, and beef cattle.
Okay, that makes sense. I don't have to ask how that path to becoming a mycologist and plant pathologist happened because it seemed like you were watching crops grow and potentially some of them not grow so well.
That's true, but I also have to give a shout out to my grandfather, who was teacher, but he was also a farmer and a big naturalist. And so he had us out in the woods from an early age, looking at different kinds of plants. And we would go looking for morels every spring. And
Hmm.
there were all kinds of interesting rocks in the creek beds, and we'd find an arrowhead every now and again. He was really instrumental in me becoming more of a mycologist, probably, than a plant pathologist, although my degree is officially in plant pathology.
Foraging for wild morels and things like that. That's a nice early memory and experience that would kind of lay a foundation for
It
sure.
really is, and he taught me a lot of the trees and to be able to identify the leaves and things like that. So it was really a wonderful experience growing up there.
Was there any pressure for you to return to help with the family farm?
No, there really wasn't. My parents were pretty much on board with whatever I wanted to do with my life. They were very helpful and supportive. A little bit of pressure from my grandmother to come back and be a school teacher so I could live close by. But other than that, no, my family's always been incredibly supportive of my career.
And with your expertise, I'm sure as time passed, you could provide management strategies for certain plant diseases or at least help identify potential pathogens on the farm.
Sure, I pass along information every now and again about things they should be looking for, and most recently, tar spot of corn has been a big issue, and I warned my dad and brother who farms with him to be on the lookout for that. I don't believe they ever found it in our farm, but it's certainly in the Midwest.
So you finish up your PhD, you go to Arkansas, you do a postdoc for a few years, and then you get hired to work in Beltsville at USDA ARS.
It was initially a postdoc, and then it turned into a permanent position, yes.
As we mentioned earlier, the timing of your career lined up beautifully with the coming of age of DNA-based studies of fungi. And you did such early work on Diaporthales, Trichoderma, Cunninghamella, Stachybotrys, Phomopsis, a lot of interesting and important genera that are still relevant, obviously, today. Was that time in that kind of early molecular biology of fungi, was that more rewarding work than it is now? Obviously, the technology, the speed that we can go now is so different. But do you look back and say, the early molecular age, like there was so much that was unknown? Tell me about that.
Yeah, there was a lot that was unknown. We also didn't have all these kits
Right.
that make life easy. We made our own reagents. We had the one PCR machine that we shared for a large group of people, and we had sign-up sheets, and the instrumentation was incredibly expensive. Now we have everybody has a PCR machine on their bench, basically, and we have these kits, and we can sequence 7 genes in the time that it took to do one. So it goes both ways. It's really an exciting time right now with all the genome-based work
Mhm.
that's happening, but it was also really exciting to be on the ground floor of beginning to look at the molecular systematics of mostly plant-associated fungi.
Yeah, because I think back to even some of the pubs I use and the primers I use for the 28S region, Renner and Samuels from
Mhm.
1994. If you're working in the early 2000s and the late 90s on this, it's not long after we're developed, some universal primers were developed for some of the ribosomal regions. Obviously, the ITS primer set was a little bit earlier, but still, there was so much that we were uncovering back then in terms of molecular biology.
I came at a really good time to be able to take advantage and utilize a lot of the work that was going on at Duke and various other places where they were developing a lot of these resources that, like you said, we're still using some of them today.
I think it's good to reflect back or at least hear like your experiences from that because I think sometimes you talk to early career scientists, graduate students, undergrads, and they say, "How did they get a paper in Nature just sequencing ITS?" And you're like, "They had to manually do the PCR using water baths and all these things," and you realize how many barriers there were to getting like good sequences back then, and you realize, oh, it was a lot harder. Like you said, those kits weren't around.
My PhD thesis was based on producing one gene from one fungus-like organism. I wouldn't cut it these days, but at the time, it was a big accomplishment. We were sequencing radioisotopes, and I still have the X-ray films. I can't quite bring myself to get rid of them. I should, but I look at them and I think of all the work that it took to produce them and on how I had to read them with a ruler because there were 4 lanes for each base, basically.
That's it's wild to think of that time. And obviously, I've talked to other scientists at your career stage, and that were there on the ground floor during this golden age of molecular biology and studying fungi. And the stories are similar, where the barriers to get a good sequence it was so ridiculously high, but when you got it, it was so revealing.
And so satisfying when it was seen as and it was a really big accomplishment and something to really be proud of.
So I look over your publication record, and Clearly, the last 2 decades, you've dedicated a considerable amount of time to studying the Diaporthales, including Diaporthi, and with some 40 plus publications. Incredible. But I also know that you've worked a lot with rust and smuts. So probably an equal number of publications, somewhere around 40 or so. With working for the USDA, this has always been a question that I've wondered about. Are your research priorities set forth by USDA? How do you specialize as a USDA scientist? Obviously, you have expertise in a couple different areas, but is that driven largely by public need?
It really is mostly driven by public need and industry need, or however you want to phrase it. We have a mandate to produce systematics of plant-associated fungi or plant disease-causing fungi. And that is set by our national program staff. Every 5 years, we go through a project planning cycle and write a new project plan with new objectives. So the focus might change in terms of which fungi are important at the time. It used to be that even within the USDA, you could come and work on a particular group of fungi and spend your whole career working on that, maybe a single genus
Right.
or a single family, let's say. That's really not the case any longer. You have to be a little more of a generalist. And molecular biology has really aided that in that DNA is DNA. So if you understand how to deal with fungi, then you can generally with whatever comes in the door. But I would say that these days, rather than specialize on a single genus, you really have to be flexible and move with the times.
Was part of the reason that you continue to work on rust and smut because they were such a difficult group to work with?
Yeah, in a way, I think that's probably true. I also had a basic interest in them. I find rusts to be just really beautiful. The spores are really fantastic looking. They're relatively large, usually the teliospores anyway. And so I really enjoy looking at those. Smuts are they're a little more cryptic and hard to find, so they're often a little more challenging, but by the same token, I enjoy photography, and there's some the most photogenic of the microfungi, and so I've found it really fun to do digital imaging and them over the years.
Yeah. I've really haven't devoted much research time to rusts, but I've spent a lot of time looking at them and photographing them. As a forest pathologist, I spent a lot of time teaching students about demicyclic rust and heteroecious macrocyclic rust. And I too am like just so fascinated with their complex life cycles and also just blown away by the fact that like some of the pioneers of our field figured out rust life cycles at a time when how the heck they connected it without DNA and all this. It just blows my mind.
No, it's true. I just, in my office, ran across my copy of Arthur. I don't know the title offhand, but his 1930s version of Rusts. And he was amazing in terms of being able to figure out these connections and names and species. And the fact that I still go back to that book. How many years later? 90 years later or some such number.
Right.
Yeah, it's amazing what he was able to do.
Absolutely. I summarize the fact that you have a considerable amount of expertise in the Diaporthales and also in rusts and smuts. If you were told tomorrow that you can only work on one of those groups, which one would it be?
That's an interesting question I've never really considered before. I think it might be rusts because I like to go collecting them and they are so photogenic. They are challenging, of course, in the lab in terms of DNA, but in terms of what I would like to spend time going collecting and working with microscope anyway, I think it would be rusts. If I were doing exclusive molecular work, it probably would be the Diaporthales because they grow like weeds in a lot of cases. And it's always easier to work with something you can grow in culture when you're doing DNA work with them. I don't find them as attractive, but they're certainly more tractable.
Absolutely. And this brings us to the perfect crossroads where we talk about your lab's role in overseeing the U.S. national fungus collections. And we just spent a few minutes talking about rusts and collecting them. And just to remind our listeners out there, rusts are obligate biotrophs, which means they keep their plant hosts alive for as long as possible while they are able to complete their own life cycles. And they can overwinter off the plants, but when they're in the active growth phase, the plant needs to be alive. As such, really the only way to archive rusts is to maintain plant vouchers with the rusts on them, the kinds of accessions and the kinds of collections that would be deposited in the U.S. National Fungus Collection and other collections. Is that right?
Yes, that's absolutely true. Herbarium collections are just absolute treasure troves of fungi and other organisms too, but certainly fungi are what I'm most familiar with. Rusts and smuts and all sorts of obligate parasites that we would have no other access to had someone not bothered at the time to stop, collect it, put a name on it, and dry it down and put it in a collection somewhere for future use. And at the time, a lot of people, a lot of scientists were doing this. They had no concept of the things that we would be able to do with these specimens now.
Absolutely. And just to further clarify this for listeners, is that mycologists and plant pathologists and other plant pathologists that study other organisms, obviously we're talking about fungi today, but there's bacteria, there's other phytopathogens. But given Lisa's expertise, we're talking about fungi here. If you can culture a fungus, then you can deposit a strain of that fungus in a collection, including USDA collections like an RSF, which is for entomopathogenic fungi. But if you can't culture it, you have to deposit a voucher specimen that maybe include both host material, plant host, and the fungus sporulating atop that, whether we're talking about a grass head or pine needles, various different hosts. But a lot of those are deposited in the U.S. National Fungus Collection. And a lot of those, if I understand, are typical of what we see in an herbarium for plants. It's a flattened sample, but there's other ways to store these things. Could you tell the listeners the kinds of ways that samples are stored in the U.S. collection?
We don't have so many that are actually flattened and glued to a sheet of paper, special archival, acid-free, etc. paper. Most of the things that we have are stored in what we call packets. So these are little paper envelopes that have labels attached to them that say what they are or where they were found, when they were found, who found them, and who maybe came along and made the identification. But it could be dried leaves in the packet. It could be, as you said, grass seeds, blades of grass. It could be twigs. We even have larger pieces of wood, could be bark. It really could be any plant part. But typically, as I said, they're stored in packets or boxes, not mounted directly to a sheet.
Sure. And as a forest pathologist, I know for a fact that some of the samples for tree pathogens are actual stems or pieces of bark with fruiting bodies on the bark, things like that.
We used to have a bandsaw that we would use to cut specimens down so we could fit them in the collection. No longer have that, but we do occasionally have to trim a specimen to be able to fit it in a box for storage.
Correct me if I'm wrong here, but if I were a researcher wanted to deposit a viable culture in a culture collection, I could also deposit field collected plant materials into the U.S. National Fungus Collection that complement that living culture that will be maintained in perpetuity elsewhere.
Absolutely. And I want to give a big shout out for you to say that, because a lot of things that people are working on, they get stored locally in their own refrigerator or an envelope in their desk drawer, and they never see the light of day after somebody's done working on it. We really advocate for people to deposit their voucher specimens because, just like I said earlier, we don't know in another 100 years what use people may be able to make of these and what sorts of scientific discoveries could be made utilizing things that we're collecting today. So we really advocate the deposit of a living culture with a culture collection and either dried material from that culture or the original plant material from which that culture was isolated with a herbarium. And of course, we like to have them with the U.S. National Fungus Collections, but there are lots of herbaria or fungaria out there where people could deposit just as easily.
There's some 1 million specimens in the U.S. National Fungus Collections. Is that an accurate number?
Yeah, it is. During the COVID pandemic, we really went to town entering data for the backlog of things that we had. And so we now have approximately 950,000 specimens that are databased in our collection. We have a lot of boxes of things that we have yet to go through because many universities are no longer maintaining their herbarium collections. We have inherited a number of them as a place of last resort for scientists or universities to put this material. So we have a number of boxes that have come from various other institutions that we're still working on getting in our database.
I'd like to talk a little bit more about the 1 million specimens that you currently have. During the COVID, you were able to catch up and digitize a lot of them. Presumably, that means that they're searchable through MyCoPortal.
That's true. We were able to digitize the labels for the little over 100,000 of the things that we had not put in the database yet. It was really a fantastic thing that we were able to do that.
One of the things that I think about is that with changes in administration, priorities change, how broader general public value scientific collections change. How stable is the U.S. National Fungus Collection? Is something we have to worry about us losing that resource? Because it's something I think about. How do we ensure that the next generation of scientists will be able to use and appreciate the U.S. National Fungus Collection, nematode collections?
We work very closely with the U.S. Department of Agriculture's Animal Plant Health Inspection Service, who is a heavy user of our herbarium and collections due to their need to identify fungi that are coming in through the ports,
Right.
either to allow them in or deny them. Within the government, I think there's certainly broad-based support for maintaining the collection. I view my role now is to make sure that there's a stable succession plan within our lab group so that someone else can pick up where I leave off. But yeah, I feel like we have really good support.
And of course, I think there's bipartisan support for those kind of efforts to help identify potential new pathogens that could impact soybean or corn or oak trees.
Yeah, and that's true, and that's one of the USDA's major missions, which is to guarantee a safe and secure food supply for both the United States and really the world.
Digitization is a big part of what's been happening in the National Fungus Collection and nematode collections in terms of getting all these records so that they're searchable to any potential user. You probably do a lot of formal loans. Is that part of what you do?
Yeah, absolutely, we do. We send a number of loans out. I probably couldn't tell you how many offhand, but we send quite a few every year to researchers all over the world. So it's not just domestic users. We send loans to researchers in Spain, in Sweden, in a lot in Europe, in Asia, wherever.
With regard to the collection, I think that there's a tendency when someone hears the word herbarium or fungal collection, they think about something in the past. Oh, a long time ago, a bunch of people collected a bunch of stuff and put it into one central facility. That's the collection. Yet that's pretty far from the truth because clearly there's people depositing specimens in 2024 and in recent years.
We still get a fair number because if you want to describe a new species of fungus, one of the requirements is that you deposit a voucher specimen to serve as a type. So we're the repository for a lot of type specimens. Molecular biology has really expanded our ability to distinguish species from one another. So there are a lot of so-called cryptic or morphological species that are being split into multiple species now. So each one of those needs to have a voucher specimen deposited. So we get a lot of those.
In February of 2024, the scientific community was shocked following the announcement that the Duke Herbarium, one of the largest herbaria in the country, houses an equal number of specimens to the U.S. National Collection, was shutting down and having its plants and other collections relocated in the next few years. As someone who is a research leader of a group that oversees the U.S. National Fungus Collection, what was your reaction and your kind of research group's reaction to this? And maybe you can help explain to listeners why that's such a huge deal.
Yeah, we were shocked, absolutely shocked, because Duke has such a long history. North Carolina in general, there's a long history of mycology and botany going back a couple of hundred years even. And they've had such a strong mycology program there for decades. We were all really shocked about that. have not heard recently what the final plans are, but it's certainly a huge blow to the research community in terms of what sorts of specimens and things will be available in the future. Depending on where they wind up, there's no guarantee that they'll be easily available, let's say, to researchers.
Right. And I think it's important to clarify something here, that just because we digitize a specimen, even if we take high resolution photographs of it, that you can blow it up and put it on the side of a skyscraper and see the details, it's not the same as actually having the physical specimen. And the idea by administrators, and I'm not saying this is what Duke administrators said, I'm just general people that don't understand the value of physical specimens, is they think if you take a really high resolution photograph of every side of it, you could just throw it away because you have evidence of it in a digital format. But that's clearly not the same thing.
Concepts of species have changed a lot over the years, and it's being able to physically examine, to look at the microscopic parts. With plants, you certainly might be able to blow it up, the image, to be able to see the parts you need to. With some fungi, it might also be possible. A lot of fungi are microscopic, and you're never going to have the resolution that you need. And again, I go back to this point that you don't know what sorts of things people will be able to do with these physical specimens in 100 years or longer, or really even maybe 20 years from now at the pace things are moving. Scientists who were collecting in the late 1700s, early 1800s certainly had no understanding of DNA and how that could be used to enhance species determinations in the future, or look for genes that might be of importance for various reasons. People are doing a lot of genome sequencing from dried material these days. It's just the thinking forward aspect of this that is really troubling, that these things may no longer be available for future work, and who knows what kinds of discoveries could have been made.
Absolutely. I think one example that I think about a lot is some of these endophytic fungi that produce interesting secondary metabolites, Paraglandula, epicloe. These are endophytic fungi that rarely sporulate outside the plant, so they are basically contained within. And only recently, in the case of the morning glories, I think after 2000, was it discovered that those alkaloids were produced by a fungus inside the plant. And it was only after germinating those seeds in the presence of a fungicide that cleared the plant of the fungus and therefore didn't produce the compound anymore. The point of this is that not only by having the physical specimens can you stain for fungal tissue inside the plant stems, but some of the secondary metabolites that are produced have a longer shelf life than maybe even the DNA of the organism. You can chemically test some of these physical specimens for secondary metabolites produced by a fungus or produced by the plant that you can never do by just having a digital photo of it.
Yeah, absolutely. We have a researcher in Europe who has regularly requested specimens from us. That's what he's looking at, secondary metabolites.
Mhm.
And he's been incredibly successful at figuring out what's there and what they might be doing and what effect there may be on the plants. So it's really amazing.
One of the frustrations of being a mycologist and plant pathologist in modern times is to see these kinds of collections devalued or boiled down to a price tag. Oh, it costs this number of dollars to maintain this building, the air conditioning, and the certain storage conditions. And that can be a little frustrating. It's probably even more frustrating for early career scientists that maybe want to pursue taxonomy or systematics. What advice might you have for someone that's interested in the kind of work you do, but also leveraging physical collections? knowing that we're moving into an environment where these collections themselves are so undervalued or devalued?
Yeah, that's an interesting question. I'll answer it by telling you how we train people who come to our facility for postdocs or internships or things like that. We try to give them a well-rounded view of everything. So they may be doing molecular work in the laboratory. They may be helping out in the herbarium. They may be working with our nomenclature specialists on how you go about naming fungi. So we try to give them that experience that they don't necessarily get at a university where the focus may be purely molecular.
Mm.
So we try to implant an appreciation for the herbaria, the collections, the old-fashioned, if you will, taxonomy of things. So that's how we're handling it. Someone who is at a location where there isn't maybe a strong collection support for strong collection, or maybe really isn't a herbarium anymore. I just would, recommend that they go outside now and again and try to get an eye for finding things. Because once you start looking and once you know what you're looking at, fungi everywhere. And it doesn't have to be just fungi, plants, animals, insects, anything. I really encourage graduate students and new scientists to go outside and get an appreciation for what's in nature and for how they might want to preserve access to the things that they're seeing.
Yeah, that's great advice. And it flashes me back to your story about growing up on a farm and your grandfather bringing you out to collect morels. I think we're all born with a sense of curiosity, but that becomes dampened over time if it's not fed or if it's not cultivated in a way. And it's really important to feed your own curiosity by going out and observing things. And it doesn't have to be what you're studying. It's just trying to find inspiration in the natural world around you so that you can then go into the lab and bring that inspiration forth to the bench.
Yeah, absolutely. I know a lot of mycologists who are just tremendous bird watchers. They'd love to go out and look for and document the birds they see. Others are equally excited about going out and photographing insects. So it really is a matter of getting out there. One thing I think that's been really helpful in this regard is some of the apps that you can find on your phone where you can take pictures of things and upload them. And there's like a community of people out there who can help you identify things, or you can put your own name on it or whatever.
Mm.
But I think that has really helped increase the level of interest and maybe even excitement.
It engages people more to observe and record what's in front of them.
Mhm, mhm. And even if it starts off with just a digital image, that's fantastic. That's documenting a thing in a place, and it's great, and if it graduates to more than that's even better. But just getting an idea of what's in the natural world around you is a great thing.
Agreed. It's been wonderful hearing about your career and your perspective on things. Thanks so much.
Thanks for the opportunity. I've enjoyed our chat, and hopefully others will too.
We've just heard from Dr. Lisa Castlebury, Research Mycologist and Leader of the Mycology and Nematology Genetic Diversity and Biology Laboratory with the USDA ARS, Beltsville Agricultural Research Center in Beltsville, Maryland. As always, be sure to check out the show notes section immediately following the episode description for links relevant to Dr. Castlebury's work and the USDA ARS. I'm Matt Kasson, host of Plantopia. Thanks for listening.